Okay, in this chapter, chapter 44, we're looking at osmoregulation and excretion. This is the 10-minute wrap-up that I will give you, and basically we're going to talk about osmoregulation. That is, how do organisms control the amount of water that they have in there. We'll talk about nitrogenous waste. Again, it's biology, so we need to talk about the different excretory systems that are there. And then lastly, we will talk about a structure, the height of evolution, the kidneys. And basically, each day your kidneys produce enough blood to fill basically a hot tub. Most of this liquid is reabsorbed back into the body, and we'll talk about that. Again, we have a balancing act. And our organs need to operate within a fluid environment, and sometimes we have too much fluid, sometimes we don't have enough fluid. In osmoregulation, we're going to regulate the solute concentrations, and that is balance our gain or loss of water depending on what our environment is. In excretion, we see the elimination of basically nitrogenous containing waste. Organisms can cover these challenges by either conforming to their environment or regulating their internal environment separate from their external environment. And basically what we want to talk about is how do we get rid of ammonia. Ammonia is basically a waste product that we see from dealing with amino acids inside of our diet, the proteins inside of our diet. Ammonia is relatively toxic, but it's really cheap to make. Uric acid is less toxic, but it takes a lot of energy to make. Ammonia takes lots of water to excrete, and uric acid does very little water. So we would say, just on average, that an organism that has excess water will produce an ammonia waste and get rid of it by urinating a lot. And an organism that does not have very much water will produce uric acid. An example of that would be birds, and we'll talk about that a little bit later. Okay, so let's go on. Osmolarity, we did talk about this during lab in class, and so we have a hyperosmotic environment, which is a solution with greater concentration of dissolved substances. Hypoosmotic is a solution with lower concentration of dissolved substances. And so when you have a semi-permeable membrane, we see that water flows from an area of high concentration, where it's mostly less solvents, to low concentration, where there's more solvents. So it's very critical that we know and kind of understand that. In isoharmonic environments, we don't really have any movement. It's relatively rare. We're always going to have symptoms. The main system that we're going to look at in basically in mammals and in birds is basically a tube-like system, where basically we have the blood flowing through a filtration device, a glomerular apparatus, and then we have tubes that can either allow reabsorption of the dilute liquid to occur or secretion to occur, and then finally the production and storage of urine in something like a bladder. Again, in filtration, what's happening is the pressure is increasing, the fluid is squeezed out. Again, we just see liquid coming out here. We don't see any formed elements like red blood cells or white blood cells. Again, through reabsorption and secretion, we can control the epithelia of this tube and control what cells get in and what cells come out. So again, life evolved in the ocean, and basically the ocean is pretty constant at the amount of salt inside of it. Again, when animals went on to land and when they went on in freshwater environments, these changed drastically from areas with very little water to areas with lots of water. And so organisms that live in these environments need to be able to handle these particular environments. The earliest strategy that we would see that many of our early animals have is just basically by conforming. That is, the osmolarity on the outside of the animal is the same as the osmolarity on the inside of the animal. Classic conformers. Not very challenging. Again, nitrogen is basically usually let to dissolve right out of the organism. However, when we started going into living things, that is, started going into freshwater environments, we had a problem. That water constantly leaked in. And again, we looked at this during lab, where we would see something like a paramecium would collect water. Every once in a while, that vacuole would constrict and just basically pump the water out. So it's kind of like being in a lifeboat that has a leak in it. As long as you can bail out the water quicker than the water comes in, you will be safe. Okay. But again, And when you have a multicellular organism, this is a little bit harder to do. Again, the flatworms, Palheminthes, use something that's kind of interesting. So they don't want to fill up with water, so what they have is a little tube system, and basically what they have is this water is able to be filtered and leak through. Lots of water leaks into the planaria and lots of water leaks out, and so basically they have a tube-like structure that goes throughout this organism that basically filters out that extra water. Again, the annelids, or these are organisms with sequential areas. In each one of these, you'll have what's called a metanephridium, and we'll talk about these in the kidneys. But basically, this is a structure that takes in fluid from the cell, and that fluid is modified. If you need to get rid of excess liquid, it will do it. If you need to get back in. Again, in all these organisms, for the most part, nitrogen is excreted as ammonia. These are very wet organisms. They grow in wet environments. However, things like insects can't necessarily do this. And what we see in insects, and again, we saw this in a dissection of the cockroaches that we did in laboratories, that we have these Malpighian tubules. The Malpighian tubules take in this liquid, and that liquid is actually absorbed back in through the gut and then excreted out in the urine. And again, so you want to eliminate the waste, but you don't want to dry out. Again, there is no filtration step inside of here, and just salts and water and everything else move through those Malpighian tubules directly, and then they're reabsorbed basically in the hind gut and in the rear gut in order to get rid of them. Sorry, it's the 11:30 samba. And so a pretty interesting system. Again, in this system here, we see a lot of times a reduction in water, and so these organisms will produce uric acid. Now, in vertebrates, and higher vertebrates, specifically humans, we see the filtration and reabsorption process happens in a particular structure that's called the kidney. Now, the kidney is pretty interesting in that every drop of blood passes through your kidney roughly about 200 or 300 times a day. Roughly about 20% of every heartbeat actually goes through your kidneys. And so if we look at the structure of the kidneys, here is the descending aorta, and the descending aorta has a large tube. This the renal artery, roughly about 20% of it goes into the kidney, and the renal vein actually comes back into the vena cava. So there's lots and lots of kidney activity that's going on. And again, the purpose of the kidneys is to eliminate urea, eliminate waste products, to balance the water inside of your blood, and also to regulate blood pressure, and that is the amount of blood that you have. Again, how does the kidney have to do with kidneys? Is that, like I said before, about 20% of every heartbeat goes into your kidneys, so it's very critical. The kidneys play a very critical role in the circulatory system, and they're very important. Know the structure of the kidneys. We'll talk about that. I want to go on real quick. This structure here that we have, the glomerular apparatus, or the glomularius, or the Bowman's capsule, is responsible for filtration. So what we see is we have a capillary bed inside of there, and fluid leaks out and is collected in this bulb-like structure. Now this is going to go into a nephron, and there are hundreds of thousands of nephrons inside the kidneys, lots of these nephrons. And what we see in the nephron, we have certain portions of this nephron. We have the proximal tubule, the distal tubule, the loop of Henle, and then the collecting duct. And we'll talk about what those do relatively soon. But again, you do need to know what these structures are and kind of how they particularly work. Again, just kind of really quick to show you what's kind of going on, is that we have the Bowman's tube, the proximal tubule, the descending limb, the ascending limb, and the distal tubule and the collecting duct. And you need to know what all of those do and how they relate to one another. Okay. So again, if you mess with blood pressure, if your blood pressure gets too great, you see more leakage in the Bowman's capsule. If the blood pressure is lower, you see less leakage in the Bowman's capsule. And again, here's a little video on that, but I'm not going to go into that excretion through the kidneys. The ocean bony fish, basically, fish gain a lot of water through basically drinking seawater, and they have osmotic loss through the gills and other body parts, so it's an excretion of salt from basically the gills. They produce a relatively small amount of urine. They don't lose much water, and they don't gain too many salts because they're able to get through it through their gills. And so lots of secretion to get rid of salts done at the gills. Very few nephrons and glomeruli. Freshwater fish are a little bit different, and they have basically fresh water coming into them, and actually structurally they have relatively large kidneys, and so they're going to have lots of absorption to keep in the salts, and they have lots of nephrons, and they produce lots of ammonia. Sharks, on the other hand, are a little bit different in that they are an osmoconformer, and they will produce uric acid in order to increase their concentrations. Again, kidney rats do very, very little drinking, and so they need to reabsorb lots of their materials. So they have very large loops of Henle. Again, in water balanced, the rats actually get the vast majority of their food through their metabolism. We human beings get relatively small amounts of our liquid through metabolism. Birds, especially seabirds, are a little bit different. They have a problem of a little bit too much salt. And so they can eliminate nitrogen through uric acid, which is a paste. It's not urinary, and they have activity glands that secrete salt, and they reabsorb the water through the cloaca. Now, kind of get an idea of how the different nitrogenous wastes are eliminated. Most aquatic animals actually will produce urea. Most animals will produce urea, and then animals that are in very dry environments, or like animals or like birds, will produce uric acid. So that's very, very important. Again, know the parts of the nephron and know what they do: filtration, reabsorption, secretion, and excretion, and know kind of where they are at. Again, the Plahomethes and Arcadia and Arthropodia, which again are organisms with the bilateral area, they have a different system. A really different system, and we've been talking about these, the tardigrades actually deal with kind of loss of water by actually forming a resting structure that is basically dehydrated. Again,